Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Optimizing Drug Loading in Amorphous Solid Dispersions, Maximizing Bioavailability while Minimizing Pill Burden. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and your comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit your questions as well via the comments tab. At this time, note that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank AustinPx, who developed the content for this presentation. AustinPx Pharmaceuticals and Manufacturing is a contract development and manufacturing organization providing analytical and formulation development services and cGMP manufacturing for small molecule drugs. AustinPx specializes in phase-appropriate development strategies, speed to clinic and market strategies, and bioavailability enhancement of poorly soluble molecules, including their next generation amorphous dispersion platform, the Kinetisol technology. At this point, I'd now like to introduce our speaker for today's event. Dr. Dave Miller currently serves as the Chief Scientific Officer at AustinPx, where he leads the pharmaceutics and analytical development teams and oversees the application of the Kinetisol technology. Dr. Miller specializes in formulation and processing technologies for improving oral bioavailability and of insoluble small molecules. So now, without further ado, I'd like to hand the presentation over to our speaker, Dr. Dave Miller. You may begin when you're ready.
All right, thank you, Ryan. Thanks for the introduction, and thanks everyone for joining us today. Really pleasure to be here once again talking about amorphous solid dispersions, particularly optimizing drug load towards the aim of maximizing bioavailability while ultimately minimizing pill burden. And so let me click into the screen. There we go. So I'm going to start the presentation just getting right to the point of a very recent case study that we've encountered with one of our clients, and that really demonstrates the balancing act that a formulation scientist does when considering drug loading against bioavailability and pill burden. In this case, we want to establish why more is often worse. We have a lot of clients come to us and their aim is maximize drug load. They're thinking ahead towards pill burden, thinking less polymer dilution in the final dosage form will result in lower pill burden. But this case study is a great illustrator of how that thinking is actually backwards. That higher drug load actually leads to greater pill burden. So this client came to us with a 50% drug loaded spray dried dispersion in a tablet, and really was somewhat satisfied with that formulation and had carried it through into late stage clinical studies. They were curious about Kinetisol and what it might be able to do for product enhancement. This is a very collaborative client that allowed us to explore the formulation space to find an optimum polymer and then optimize the drug load. It's not always the case. Some folks come to us and they really want to stay within a very well-defined formulation boundary, but we can leverage the power of our Kinetisol platform more if we have the freedom to explore the formulation space. So in this case, client allowed us to do that. And what you see on the screen is kind of the end result of that study. It culminated in a primate PK study of what we considered our lead Kinetisol dispersion versus their phase 2 spray dried tablet. And what you see is we found an optimum polymer, optimized the drug loading, and in this case, 15% drug load was deemed to be most optimal. And that's, in a range that I think a lot of folks would say is a little too low. But what this result shows is that it's not. Because when we went into primates with our kinetosol tablet with a 15% drug-loaded ASD versus a spray-dried tablet with 50% drug-loaded ASD, what we saw was the kinetosol product achieved a 5x increase in AUC versus the SDD. So even though the SDD had a higher drug load, about 3 1/3 times higher than the kinetosol formulation, kinetosol was able to achieve 5x the AUC. So what that means towards pill burden is that KSD requires a 1/5 of the total dose of the SDD product. So 100 milligrams of the kinetosol dispersion is equivalent to about 500 milligrams of the spray dried dispersion. So by selecting the most, judiciously selecting the best polymer and optimizing the drug load to increase the fraction absorbed, we were ultimately able to reduce the pill burden. So, starting off the presentation with really putting the punchline first. This is really key. And I'll supplement this, looking to the literature for where this has been taught for, almost 2 decades now in terms of ASD drug loading and how you can get into diminishing returns as you escalate drug load. And I use examples from the Bend research papers that came out in 2008 and 2009, Cortolo et al., Friesen et al. Those are really seminal papers in the space of amorphous solid dispersion. The group in Bend, working with Pfizer, really laid the foundation of amorphous solid dispersions very quietly for a couple of decades until they published these papers. So some of the best examples and the foundational understanding of ASDs are demonstrated in these 2 papers. And if you haven't read them multiple times, you probably should if you're working in this space. So I like to point to these examples, these 2 figures that are on the screen. What you can see on the left figure is escalating drug load from 25% up to 66%, how the concentration of dissolved drug decreases dramatically as you go up in drug loading. So again, in that case, more drug in the ASD is actually less in terms of free drug available for absorption and what can actually be absorbed in a patient, which ultimately equates to the fraction absorbed. So a 25% drug load ASD is more efficient in terms of fraction absorbed, substantially more efficient than something at a 60% or much higher drug loading. And a slightly less dramatic, but a similar trend is shown on the right. Again, a different compound in the same polymer showing decreasing dissolved drug with increasing drug load. So this needs to be considered. The amount of drug available for absorption, fraction absorbed, needs to be balanced out against the amount of drug you can actually fit into the final dosage form to find that optimum. So that's the balancing act. And then what can't be understated, really the crux of the first example that I showed, is that polymer selection is critical. And these, again, figures from the Cortolo paper from back in 2009, 3 examples, 3 different drugs showing how polymer chemistry dramatically affects the amount of the extent of solubility enhancement and the maintenance of supersaturation, and how that's influenced by drug polymer interactions. So the polymer chemistry is what governs drug polymer actions in situ, and like I said, it influences the extent and the duration of supersaturation. So what we call the parachute effect, which I'll get into a bit more for those that don't know or aren't that familiar with the spring and parachute analogy. I have a figure that will speak to this in just a moment. But the parachute effect is the ability of a polymer to sustain supersaturation. And that can vary significantly. For a given compound, it varies for the, different polymer chemistries will have different effects, as you can see with these figures shown above, but also across compounds where the functional groups, the character of the molecule is quite different. The rank order of polymers and how they interact with the drug can change very substantially as you look across chemistry space. So for each drug, there's not really a template formulation. It's important to evaluate the drug polymer interactions on a per molecule basis to find the optimal polymer in terms of interaction in situ that lead to better bioavailability, better fraction absorbed. And in this case, HPMCAS is often the best, but not always the best. And that's why here at AustinPx, we really like to take time to judiciously evaluate drug polymer interactions to select the best polymer in terms of biopharmaceutical performance, and then obviously also checking the boxes for chemical compatibility, physical stability, and ultimately manufacturability. So I came out of the gates hot with some good examples from our own experience and from the literature. And now I'll go through the rest of the presentation, but I want to call out the key themes. So first theme, maximizing absorption starts with selection of the right parachute. So it's important to pick an ASD platform that offers the broadest polymer options. The more options you have to formulate your insoluble molecule in terms of the polymer carrier, the more likely you are to find the best formulation for your drug. And also, as I've already discussed, drug-to-polymer ratio has to be optimized, not necessarily maximized. And when folks come to us and the idea is maximize drug load, put as much drug as you possibly can into the ASD, as much as you can before the drug starts recrystallizing, that often is counterproductive. So more drug in that case can mean less absorbed due to phase separation and precipitation in situ. Further, increasing the drug load to try to reduce pill burden can backfire, as we saw in that first example. They increased the drug loading to 15% to 50%, and required a much higher dose than if they were to decrease the drug load down to 15% in the correct polymer. Finally, technology selection, as I will cover in depth towards the end of the presentation, is really critical to maximizing performance. So technology really determines your formulation design space, which excipients you can use to formulate the product, and also often overlooked, but now becoming more and more apparent in the literature, are how particle properties are impacting performance, and there'll be a lot more on this later. Finally, tablet level ASD loading is a critical lever. It's not just about drug loading in the ASD, it's also about how much ASD you can load in a final tablet while maintaining target tensile strength and disintegration properties. And as many of you on this webinar know, spray drying often limits loading capacity of the SDD in the final tablet because you need to dilute the SDD prior to dry granulation roller compaction and then dilute once again moving into tableting. So spray drying is at a disadvantage in the amount of external excipients one has to apply to the ASD in order to achieve an acceptable tablet. So quickly, want to sort of center the presentation for those who maybe are not as well versed in the space is the problem of insolubility and how insolubility prevents therapy. Using this maximum absorbable dose concept, it's a conceptual linear equation that helps to estimate how solubility limits the maximum absorbable dose. And so I won't belabor the equation too much, but I will focus on the figure on the right that really demonstrates that when you're dealing with an insoluble drug, something with a solubility in a range of say 1 to 50 microgram per mil, it's very limiting to the dose that you can administer and be absorbed and achieve therapeutic efficacy. So when we're in a range of low solubility and high dose, as many compounds in some of our favorite disease therapeutic areas are, this requires formulation intervention. We have to achieve solubility enhancement through formulation technologies in order to enable therapy with these molecules. So how do we enhance solubility of a hydrophobic drug? We'll look at this sort of conceptually through the dissolution process and understanding that for hydrophobic drugs, that the dissolution process is not spontaneous, right? You've got this stable crystal lattice, you've got to dissociate the molecules from each other. That requires a certain amount of energy. Then there's this cavitation within the aqueous solution solvent that you need to create that requires energy. And then there's the energy of hydration to hydrate these individual molecules. And once you've got these molecules in solution, if you're able to overcome and if you go beyond the equilibrium solubility, you'll get into the situation that we're describing below, where you've got a molecule in a medium where it's not particularly happy. It's a high energy state, and it'll look to find itself in solution and nucleate and propagate the particle growth, right? That's what happens when we supersaturate an insoluble drug in the media. So how do we improve accelerate and enhance this process with formulation intervention? Well, there are equations that govern supersaturation, nucleation, particle growth. I won't belabor this to any great extent, but just know that it's foundationally understood. We know how formulation factors impact supersaturation, and then how they can impact nucleation and particle growth. The take-home message is that concentration-enhancing excipients disrupt the nucleation and particle growth process. They enhance supersaturation, they disrupt the processes that accelerate nucleation and particle growth. And so we'll illustrate this with going back to that figure and pictorially describe this parachute effect and the function of concentration enhancing excipients. So now when you formulated an insoluble drug substance in such a way that it's able to supersaturate, but now instead of the drug being sort of naked in solution, you've got excipient materials that can interact with the drug and prevent it from finding itself nucleating and growing. And so, pictorially, we're representing that. So favorable drug excipient interactions sort of reduce the energetic barrier towards dissolution supersaturation, and then reduce the energetic driver to go back the other direction for nucleation and crystal growth. And so at the heart of this presentation is that you want to tune the X, the number of molecules in solution, against Y, the excipient concentration. You want to tune that to optimize your Z, your drug excipient interactions toward, and you want optimize it so that you can achieve maximum bioavailability with minimum pill burden. Again, primary theme of this presentation. Okay, I alluded to this earlier. I feel like if you give an ASD presentation these days, this is almost a requirement. You have to show this figure. I love it. It's really describes the heart of supersaturating delivery systems and the impact of excipients. So you start off with a crystalline drug's dissolution profile. It's an insoluble material, slow to dissolve, poor, low equilibrium solubility. So you're sitting here in the situation where the crystalline drug dissolves up to CEQ, and that's well below what you need to achieve absorption of a higher dose and ultimately therapeutic efficacy. So you reduce that energetic barrier of dissociating the crystalline particles into its constituent molecules. So you create an amorphous form. You break through various processes. You already disrupt the crystal lattice so that you can get to a disordered form of the drug that's more likely to spring into solution. So here's where you go. You got an amorphous spring, and it springs into solution, and you get, for a brief period, very high apparent solubilities, and then without any excipients in solution interfering with nucleation and crystal growth, the spring rapidly crashes back down to the crystalline drug substance. And this usually happens pretty fast so that the amorphous payload is not available to be absorbed where it's needed, which is primarily intestinal tract. This all happens in the stomach where very little absorption happens, supersaturates, crashes out, and you're back to where you started from. So the amorphous form doesn't really do you any good without a parachute. And so here's the concept of the parachute. So with the parachute in place, once again, you can achieve higher apparent solubilities than the pure amorphous form in some cases. It promotes a more energetically favorable environment for the drug to enter solution. And then, as I've shown before, it interferes with drug-drug interactions, either sterically or through chemical interactions, intermolecular interactions, and delays nucleation and crystal growth so as to maintain supersaturated concentrations, free drug available for absorption for a duration, ideally, that spans at least the intestinal transit time. So you present the drug in a form that's more available for absorption, maximizing the fraction of drug that can be absorbed and ultimately achieving therapeutic efficacy at the lowest possible dose. So this is all conceptually understood. So let's talk about historically all of the various approaches that we've used as formulation scientists to enhance the solubility of a drug substance. So the simplest way to do this, simplest supersaturating delivery systems are these liquid forms, like non-aqueous vehicles, like lipids and PEGs. And then you can take that one step further and go into emulsion formulations like self-emulsifying drug delivery systems, taking lipids, incorporating surfactants so that you can control particle size once it's administered. So this is a is a great tool, and it works a certain amount of time, but what we often see is that their applicability is limited by the drug's solubility and these vehicles as well. If it's high dose, it has to have very high solubility in these carriers in order to accommodate the drug's dose within a reasonably sized soft gel capsule. So oftentimes these liquid forms don't apply because the drug solubility in these vehicles is also limited. And then when you look at solid forms such as crystalline salts, co-crystals, polymorphs, nanoparticles, microcrystalline dispersions, again, these crystalline forms limit the amount of apparent solubility improvement that one can achieve. It's pretty much constrained to this somewhat to the drug's baseline equilibrium solubility. So these don't really apply when you think back to that maximum absorbable dose curve and you need solubility enhancement on the order of, an order of magnitude increase or several multiples. These crystalline approaches, these solid form approaches typically don't work well. So that leads one to an amorphous solid dispersion approach, where you take the drug, molecularly disperse it in a polymer, and the polymer stabilizes the drug physically, but also promotes supersaturation and maintenance of supersaturation. And it's perhaps the most applicable form in terms of there's several different technologies to form solid dispersions, several different polymer chemistries available. So it's a very broad platform. It can apply to most drugs, or almost all drugs, independent of their properties. So a very powerful tool for formulation scientists working with insoluble drugs. So quickly, I'll go through the anatomy of an amorphous solid dispersion. I think most of you are very familiar with this, but we'll calibrate the audience here. So what it is an API that I'm not even going to say ideally. These days, it's molecularly dispersed within a polymer matrix. This is usually done by fusion-based or solvent-based processes, basically disrupt the crystal lattice, distribute molecules in a polymer matrix. So the most common ASD compositions are simple binary drug polymer formulations. In some cases, as you'll see with commercial products, you'll see drug polymer surfactant. And then as we're moving forward, so binary, we're kind of considered, first generation ASDs. The ternaries are kind of second, third generation ASDs. I think in 2025, we're moving beyond ternary. We're going into multi-component ASDs. These are more rare, especially commercially, but they're becoming more common in the development space. They're very technology dependent, as I'll describe in a minute, but they can be very effective for maximizing bioavailability, stability, and manufacturability, as well as achieving very powerful drug product IP, and more on that later. But yet you use a multi-component system not only to improve the biopharmaceutical performance, but also to ensure physical stability, chemical stability. You can modify release with multi-component systems, and you can really maximize the drug load while maintaining performance and stability using a lot of different materials at once. So the role of the polymer in an ASD is to, first and foremost, you need to physically stabilize the amorphous form of the drug. If it reconverts to crystalline on storage, it's not an effective product. It's not an approvable product. And then when you dose in a use environment, in an aqueous environment, the role of the polymer is, as I've already described, to increase the apparent solubility, allow for supersaturation, move the point on the MAD curve way up so that you can, the maximum absorbable dose goes up into the hundreds of milligrams to accommodate a therapeutic dose. And then once you're there, and maintain that apparent solubility for the duration of intestinal transit so that the full dose can be absorbed. And for those ternary systems, a surfactant you'll find improves wetting of the ASD, accelerates dissolution rate, but it also can act synergistically with the polymer to promote the fraction of drug available for absorption. And then, as we'll talk later, this goes beyond surfactants and into multi-component systems, which we'll get into in a moment. So insoluble drugs are coming out in waves starting in the early 2000s. We have all of these sort of historical liquid formulations, solid state chemistry, solid state approaches to improving solubility are proving to be ineffective, and ASDs become sort of the primary mechanism by which we can deliver insoluble drugs. With that, spray drying and hot melt extrusion really emerge as the leading ASD technologies. This is this is very evident in this figure that I have to apologize. To me, this figure is pretty recent, but I have to realize it's 5 years old now. I need to update this figure, but it illustrates the point that around this time, there's an explosion of approvals in terms of amorphous solid dispersion-based products. Prior to about 2007, there's sort of a smattering of some amorphous products. And then about 2007, 2008, the approvals of amorphous solid dispersion-based products really increased exponentially with spray drying and melt extrusion leading the space in terms of technologies. So it's about this time where ASDs really started to take off that Kinetisol was born. And it was born at the intersection of pharmaceutical science and plastics recycling. To put that in human form, it was my colleague Chris Bruff, who was our chief technology officer, was pioneering a novel plastics recycling technology that was born out of the limitations of melt extrusion for post-processing mixed waste plastics. So melt extrusion was very limited in its ability to take mixed trash, basically, and produce recycled products. So this technology, thermokinetic compounding, was invented to overcome those limitations. So I met Chris and this process in 2007. I was a grad student of Bill Williams and Jim McGinnity at the University of Texas. I was focusing my research on melt extrusion for the application of amorphous solid dispersions, really trying to maximize the formulation design space, increase the number of polymers and common materials that we could utilize in formulation, and the number of molecules that could be produced by melt extrusion. And I was really struggling. As many of you know, melt extrusion, we're dealing with added heat, high temperatures, but most importantly, prolonged residence time, and really significant polymer viscosity limitations. So I was really constrained on all sides as I was trying to expand the formulation design space in melt extrusion. So when I met Chris and learned about his process and how it overcame sort of analogous challenges to plastics recycling, and then I educated Chris about what I was trying to achieve with melt extrusion, we both sort of simultaneously came to the conclusion that we needed to try pharmaceutical materials in these plastics recycling machines. So we did those first experiments back in 2007 at the industrial scale. The first data sets that we generated really exceeded everyone's expectations. And as a result, joint IP was filed between Chris's company and the University of Texas at Austin for the pharmaceutical applications of this technology. And a company was spun out of the university to commercialize the pharmaceutical applications. So since those days, 2007, almost 20 years ago, the Kinetisol platform has evolved as an equipment platform. Chris's team leads the design, fabrication, optimization of the pharmaceutical equipment, and the application of the technology in pharmaceutical spaces. And we've spent a lot of time over the last 20 years really demonstrating the novel applications of Kinetisol for pharmaceutics. And then the third bucket is the manufacturing science associated with the technology. Once you've got a formulation application, it's scaling it up, tech transfer, manufacturing at a large scale. And my good friend and colleague, Justin Keene, our Senior VP of Operations, really leads the manufacturing science. So we've got 3 parallel paths that have been evolving over 20 years to the point where we are today, where the machines, the applications, and the manufacturing science are well established and mature. And it's a very valid commercial process. So how does it work? This is a simple schematic of the Kinetisol process. So what you're looking at on the screen is a cross-sectional view of the mixing chamber. So it's a cylindrical mixing chamber mounted in a vertical orientation. There's a high-speed shaft running through the center of the chamber. Protruding off that shaft, what's represented here by simple rectangles, in reality, there's a very specific geometry, are what we call the mixing elements. Inside the chamber, flush with the inside diameter, is a temperature probe that's monitoring product temperature in real time at high frequency, and that's the mechanism by which we control the process. So what comes into the process is a physical mixture of the crystalline, most often crystalline drug substance. Doesn't have to be crystalline, but that's typically what we're dealing with. A blend of the crystalline drug substance and the various excipients that compose the carrier are charged to the chamber all at once. The process is initiated. There is an example let me get my pointer. There's an example of the temperature profile in the right. So the process from initiation to completion takes about 12 seconds. So what comes in is a powder. What comes out at 12 seconds is a molten mass of molecularly dispersed drug within the molten excipient carrier system. That molten mass at 12 seconds ejects from the chamber simultaneously. It's quenched in a compression operation where it's compressed between plates and water cooled back to room temperature. And then what comes out, I have some examples here. This is on the research scale. It's a glassy amorphous disc. It's brittle. This one happens to be with HPMCAS. It's easily breakable and thus is easily millable using conventional pharmaceutical milling technologies into a fine powder. And that powder can then be moved downstream, directly compressed into a tablet, filled into a capsule, dosed in suspension. It's very versatile. And what I'll point out about this example temperature profile, not only is it remarkable that we go from physical mixture of crystalline drug substance and excipients to a molten molecular mix, and then finally an amorphous glass within about 10 to 20 seconds. That's remarkable in and of itself. But I want to point out that this example is with vemurafenib, which is kind of a famous molecule in the ASD space, a brick dust compound that Roche commercialized using. It couldn't be spray dried, couldn't be melt extruded. They had to do solvent anti-solvent precipitation to move this molecule forward. This example shows that kinetosol makes amorphous dispersions equivalent to the commercial ASD at a temperature about 100 degrees below the melting point of the drug. So that's remarkable as well, and I'll talk about that more in just a minute. So what I said, this is an evolution of equipment, formulation platform, and manufacturing science. Well, this is the equipment part. We have two scales of kinetosol equipment. We have our R&D, our lab scale equipment. This is where we do our feasibility studies. This is where we explore the formulation space, tailor make the formulation for the molecule, simultaneously solving the bioavailability needs, stability, and manufacturability. We work this all out on a small scale on an API sparing basis. We can exhaustively explore the formulation space with a relatively small amount of API and very short amount of time. We can make dozens of compositions on this machine in a given day. It's highly efficient for formulation screening. And once we identify our clinical candidates, our formulations we want to carry into humans, we can make first in human supplies at this research scale if we need to get into clinic quickly. Otherwise, there's a scale-up event to the manufacturing scale, and that's the final scale-up event. There is no increased scale of kinetosol. There's one manufacturing scale, and that has tremendous advantages. So the scale-up is very simple. Everything remains the same. The rotational speed of mixing is reduced by about a factor of a half. So very efficient for scale up. And then when we get up to the manufacturing scale, it's a semi-automated process, just like tableting. Powder is charged from the hopper into the chamber. A shear force is applied to produce a molten mass. The molten mass is ejected from the chamber and quenched. Meanwhile, the next bolus of powder is being introduced in the chamber, and the process repeats itself. So this is perfectly analogous to tableting, where powder is charged into a die, a compression event occurs, a tablet's created, the tablet product is ejected, and then the next powder is introduced into the die. And this happens in rapid succession, so that tableting is a semi-continuous process that's happening in such rapid succession that it's effectively continuous, and kinetosol operates under those same principles. So what's brilliant about manufacturing with kinetosol, we scale up to the next scale. This is applicable for first in human, so we can make a couple hundred grams of dispersion. We got 1,000 dose units, if at minimum, and we do that by running the process for about 30 minutes to an hour. And then when product demand goes up as clinical, the molecule progresses through the clinic and eventually to registration commercialization, we don't change anything about the process except for the duration of operation. So where we were running for 30 minutes to an hour for first in human, later in the product life, we run the process for 3 shifts continuously for several days to produce metric tons of material. So there's no scale up, it's just a matter of duration. So what immediately jumped out at me when I was in my former life, when I was as a melt extrusion scientist, when I saw this process for the first time, the time under the temperature curve, or sort of area under the temperature versus time curve relative to HME. So I won't belabor this too much, but what we want to point out is that this is the same product. It's an itraconazole HPMC product. The orange line represents the commercial melt extrusion process in terms of time at temperature. And this blue curve is the equivalent kinetisol-based product. And this is the temperature profile of kinetisol. So you don't need to do the math. You can look and see the area under these two curves is dramatically different. And what that equates to is substantial reduction in the generation of impurities. So compounds that are too thermally unstable for melt extrusion are perfectly processable by kinetisol, and that's the first advantage. When a lot of companies would prefer thermal processing over solvent processing, but the issue is, as many of you know, melt extrusion is only applicable to, in my experience, about 20% of molecules. The rest are too unstable. And then before you even get into excipient instability and high viscosity that limits the design space of melt extrusion. And so producing ASDs with a minimal amount of thermal exposure is one obvious and primary advantage. The next is being able to solubilize high melting point compounds well below their melting temperatures is also unique to kinetisol over melt extrusion. And I generated this figure a few years ago just to show sort of the range of melting temperatures that we've successfully developed products in over the years. So what you see is about 3/4 of what we worked on would be considered high melt, 200 degrees C and above. And I'll point out some of these here more recently where these molecules have melting temperatures north of 300 degrees and even north of 350 degrees. So these are ultra high melt brick dust compounds. You wouldn't dream of trying to melt extrude these to any reasonable drug load, but these are routine for kinetisol. And then you look down, we have some, we've worked on some fairly low melt compounds as well. So melting temperature really doesn't impact the applicability to kinetisol whatsoever, but it definitely limits what you can do with melt extrusion. And then let's talk about the formulation design space. So there's all of these various primary carriers available for ASDs, ranging from polymeric, like non-ionic polymers, ionic polymers, and then getting into even non-polymeric materials. So, and there's various chemistries under these categories as well, and subchemistries and subgrades. There's so many polymer options out there, but yet there's very few that have actually been commercialized. And this is kind of reflected in some of our clinical stage kinetisol products. We really kind of stick to the polymers that have been tried and true. But as we progress with kinetisol and explore a wider design space, more of these polymer chemistries and their various subgrades become critical to maximizing the performance, stability, and manufacturability of all of these molecules that we're seeing. We're really, as an industry, pushing chemical space to an extreme. And as we do that, as we push what is developable as a drug, we need more flexible formulation options. And this is where we get it with kinetisol. We can use any of these polymers, combinations thereof. And going on to the next slide, it's not just about polymers, combinations of polymers. It's also, as we said before, surfactants, wetting agents. We can incorporate cyclodextrins or do cyclodextrins directly, but also looking at like chemical stabilization, putting in antioxidants, pH modifiers. There's so many different options that in terms of developing an ASD to solve, like I said, you've got these three simultaneous challenges of bioavailability, stability, manufacturability. You've got to keep all of these things in mind as you're developing an ASD, and a simple binary drug polymer system just doesn't really cut it these days, especially if you're looking for IP. There's way too much prior art. So what we do is leverage this platform that we call kinetisol and to tailor make multi-component ASD systems to simultaneously solve all these problems to maximize the performance of the ASD. And when you've done that, you've got a composition of matter that's a surprising result that stands out amongst the exhaustive experimentation as something unique for your molecule. And that's a composition that can't be duplicated, can't be copied, is patentable at the patent office, and would hold up, and increase the lifespan of the asset. So that's what we talk about when we're saying kinetisol has shifted the boundaries of ASD. We can apply it to more molecules and far wider formulation space. And this slide just illustrates some of the other advantages. Obviously, kinetisol requires no solvents, so I think we know what the benefits are there. I've already talked about minimizing thermal exposure. This slide demonstrates the much wider formulation space. And then when you talk about the mechanism of formation, as I've described, solubilization of crystalline drug in the molten polymer, it's a dissolution process. We get above the TG of the polymer through mechanical energy. We don't add any external heat. It's mechanical energy, changes the polymer itself into a more malleable, flexible, fluid-like state, and that acts as a solvent for the crystalline drug. And so we're dissolving crystalline drug within the polymer. So it's like a dissolution process where you're spinning that dissolution paddle really, really fast. So we're solubilizing that crystalline drug within seconds. So now let's look at particle properties. Kinetisol particles, pretty similar to melt extruded particles because the particle properties they're produced they're influenced downstream of ASD formation. So we form the ASD, we make this molten mass, we've got this crystalline glass, this disc that I just showed, and then we use the downstream milling technology to engineer the particles. Now that's a really distinct difference from spray drying. It's similar to melt extrusion, but it's a very distinct difference to spray drying, where the particle properties are dictated by the ASD formation process. So there's not as much control. And so they're very different. If you look at the SEMs of the two particles, it's very different. Kinetisol particles are very dense, sort of granular. Spray-dried particles, as we all know, sort of hollow spheres, corrugated spheres. So what does this mean in terms of downstream handling? Well, for one, kinetisol material coming out of the milling process has very high bulk density, typically 0.5, 0.6. Contrast that to spray drying, as many of you know. Spray-dried powder has very low bulk density, and therefore it has to be densified in order to be further processed into a final dosage form. Our kinetisol material has excellent flowability, whereas bulk spray-dried powder tends to not flow well. As I already said, we can tune the properties downstream of ASD formation. We can use milling technology, conventional milling technologies, to modify the particles as we need to optimize performance of the dosage form, as well as manufacturability of tablets. Whereas, the spray-dried particles is limited tunability because it's, inherently part of the ASD formulation process. I will talk in just a moment about the directly compressible nature of kinetisol particles, whereas, as we know, spray-dried material is not directly compressible. It has to be densified first. And then most importantly, as I will show with the case studies in just a couple of minutes, dense particles promote congruent drug polymer release. The amorphous drug substance doesn't come out of the particle until the polymer is soluble. They release simultaneously, whereas high surface area spray-dried materials, even solvent, anti-solvent particles that have high surface area, it is very common that the drug and polymer have different dissolution rates, specifically when there's pH dependencies involved. So incongruent drug polymer release, if you go back to that figure that I showed earlier, that means the drug is out in solution, potentially naked, without polymer protection, and you get nucleation and crystal growth. Whereas with congruent drug polymer release, you don't. So kinetisol dense particles, like the ones you produce with kinetisol and melt extrusion, promote congruent drug polymer release and maximizing fraction available for absorption, whereas high surface area particles do not. So this takes us to technology selection. So let's look at technology selection first based on API properties, melting point versus solubility in organic solvents. So if you've got high melt, poor solubility in organic solvents, we're in that top left quadrant up here. Kinetisol is your only option. And then as you go around the quadrants, you can apply spray drying and melt extrusion, but kinetisol is also applicable there. So now let's also look at it as thermal instability versus organic solvent issues. Now, I don't have the time to go into organic solvent issues, but any of you that have worked in spray drying know what I mean when it comes to organic solvent issues. So if you've got a molecule that's thermally unstable, and it's got a lot of issues, whether that be solids load or your organic solvent is chlorinated or it's THF or something nasty, you're sitting up here in a quadrant where kinetisol is probably your only option. And then as you go around this grid, once again, you can apply spray drying and melt extrusion, but kinetisol is also applicable. So the take-home message here is kinetisol is the only technology that's applicable across all these quadrants. So when you think about if you've got a given molecule and you're wondering what technology you should go with, you can always evaluate kinetisol. There's no limitation. You can try it. And we have the widest formulation design space. So we can take your molecule and screen it with any polymer that you should desire. Certainly all of the standard ASD polymers, but where we can get off into things like polyvinyl alcohol and cyclodextrins and things that you really wouldn't consider with spray drying and melt extrusion. So now let's look at part 2 of technology selection. When thermal and solvent property consideration. So we like spray drying because the molecular coverage is excellent. I mean, if the drug's got even a little bit of solubility in an organic solvent, you can evaluate spray drying. So it's great from that perspective, and we know this. Now, when we look at the chemical space, though, what limits what you can do in terms of formulating an ASD is that the excipients have to be soluble in the same organic solvent as the drug, and that really limits what you can do in terms of formulation. And then when you go down the list, obviously organic solvents not desirable. Scale-up complexity is significant with spray drying. Operating cost tremendous. Capital cost, just huge for spray drying. You have to build a building around commercial spray dryers. Commercial capacity, it's becoming limited. I mean, think about with the rise of GLPs and how much spray drying is being done there versus just limited commercial capacity versus in the demand of insoluble drugs. Like commercial capacity is becoming a risk for spray drying. But there's extensive commercial history, though. That's why the industry likes it. Good molecular coverage and a lot of history, right? It's low risk. But finally, energy efficiency is poor. So, melt extrusion, the biggest issue, molecular coverage is pretty poor, as I've already talked about, and the design space for formulation is pretty limited. So we look at the kinetisol, again, it's coming up green in all these categories but one, and that's the commercial history. So the take home for, at the end of the day for this is that spray drying and melt extrusion had a 25-year head start. If we were all born on the same day, kinetisol would be the go-to ASD technology within the industry, but we're behind, but we're quickly catching up, as I'm about to show. For the sake of time, I'll probably skip over this, but I've already kind of described how the scale up is very straightforward with kinetisol. We go from research scale to manufacturing scale, and that'll take us from phase 2 to commercialization. And this equipment is portable. It can be placed anywhere. The utility requirements are electricity, chilled water. Contrast that against spray drying, where you go from a Buki all the way up to a PSD 4 or 5 by the time you're at commercial scale. There's so many scale-up events, so many challenges in terms of changing particle size, how that affects downstream processing, secondary drying, roller compaction. There's a lot of challenges you face for spray drying that we just don't have with kinetisol. So now, with all that stated as background, I want to go through very quickly some of our case studies. Most of these are already in the literature, so I've referenced some of our papers, so you can go to these papers if you want to learn more. But starting out with ritonavir. So the problem statement is ritonavir is an ASD product. It's a tablet called Norvir. It's a low drug load, large tablets, 15% ritonavir in the ASD, and the tablet total weight is about 800 milligrams. Now the real problem is ritonavir is not used as a therapeutic agent directly. It's used for PK boosting primarily. So you'll find it in a lot of fixed dose combination products. So a low drug load and high external excipient requirement, large tablet, isn't really amenable to a combination product with other antivirals. So we set out to use kinetisol to improve the drug loading, decrease the pill burden of Norvir. And we did this by utilizing our platform to use, like I said, the best concentration enhancing polymer for ritonavir, and that's HPMCAS. Now, why is kinetisol unique? It's because ritonavir is thermally and acid labile, and HPMCAS is an acidic polymer. So if you try to melt extrude this composition, you'll degrade ritonavir extensively. We are able to use kinetisol to produce this dispersion because the material is only at elevated temperature for about 2 seconds, and the Tmax of the process is 90 degrees. So it's 30 degrees below the melting point of ritonavir. So that's what enabled thermal formulation. So you see, we get a perfectly amorphous dispersion and an impurities profile that, despite the fact that we are using a highly acidic polymer and Norvir uses a non-acidic polymer, our impurity profile is actually cleaner than the Norvir tablet. And when we looked in, are we truly making a molecularly dispersed dispersion in only 2 seconds? Well, we did solid state and MR analysis and proved that we had a fully amorphous system. And looking at T1 row, there's no difference. The phases it's completely single phase. a molecular dispersion. And now let's talk about tableting. So that's another key component, as I said before. You can optimize drug load, but you also should optimize the ASD load in the final tablet. So our kinetosol material is directly compressible, has excellent compression properties, which I'll get into in a moment. And we were able to increase the weight percentage of the ASD in the final tablet to about 76%. So we were able to get 30% drug load, 100 milligram tablet, and about half the size of a Norvir tablet. And you can see with good tensile strength, excellent disintegration. So now at Austin Pharmaceutics, we really believe strongly in having numerous in vitro tools for establishing biopharmaceutical performance. So we look at various biorelevant dissolution methods and also combining that with transmembrane flux to get two different data sets to select formulations. So this is what you see on the screen. On the left, the donor side, a pH change, non-sync gastric to intestinal transfer dissolution system. So you see very different release profiles. Norvir releases extensively in acid and then precipitates after the pH change. Meanwhile, our formulation is enteric based, releases to a minimal extent in acid and then supersaturates in intestinal media. And then when we look at the flux, the transmembrane flux of ritonavir from both systems is fairly similar. And then when we went into dog PK, what we saw at 2x the drug load in our kinetosol tablet, we are, based on AUC, essentially equivalent to Norvir. So by judicious selection, right polymer, optimizing drug load, we were able to get bioequivalence at twice the drug load. Okay, we only got a couple. I want to leave a little bit of time for questions, so I'll go through these next case studies very quickly. This is a pill burden reduction example using kinetosol compared to a commercial spray dried tablet. So in this case, the commercial tablet is a 25% drug load SDD. It's spray dried from dichloromethane and ethanol, so chlorinated solvents. The EPA doesn't like them. Most of the industry doesn't like them, so it's highly non-ideal. As most SDDs are all SDDs, secondary drying and roller compaction is required. The high dose tablet is very large, 1.2 grams. So spray drying was limited to 25%, but when we were working with this molecule, we were easily able to generate amorphous solid dispersions between 20 and 40%. But we started with drug product development matching the SDD drug loading at 25%. We were once again able to leverage our biopharmaceutical tools to show similar dissolution performance to the SDD. We took it through our tableting process. We were able to get 85% ASD load in the final tablet. So we were able to generate the high dose tablet with slightly smaller size, 1.129 grams relative to the 1.2 gram SDD tablet. So a little smaller tablet size at the same drug load. Again, looking at using our biopharmaceutical tools, similar dissolution and flux profiles between the tablets. And then finally, we went into an in vivo PK model and established bioequivalence of the kinetosol formulation to the commercial SDD tablet. But with that, we wanted to further reduce pill burden. And as I showed you before, we were able to produce ASDs at 40%. So let's develop a drug product at 40% and see how much we can reduce pill burden. So we were able to take this 40% dispersion with 75% ASD in the final tablet. We're able to make the high dose tablet at a total weight of 800 milligrams. So 50% smaller than the commercial SDD tablet. And when we run the in vitro with some optimization of the ASD, incorporating some other components, we're able to get, at this point, we're at in vitro, in a predictive in vitro method showing equivalence to the commercial formulation. So we're moving ahead with this formulation into in vivo studies with high likelihood of showing bioequivalence of the commercial spray dried tablet at a higher drug load and 50% less tablet mass. So moving on, I want to talk briefly before we do Q&A on the impact of technology selection. So we talked about judicious polymer selection, optimizing drug-to-polymer ratio, but how does technology selection impact performance? And I've already showed it on the slide previously, contrasting KSD particles versus SDD particles. Particle properties are critical. And we learned that many years ago working with Boehringer Ingelheim. The problem statement was, we've got SDDs that we're using for tox studies. We need a more efficient, cost-effective way of manufacturing them. So they utilized kinetosol to duplicate the SDD formulation. And so it's the same formulation, just made by a more efficient process. And what we found serendipitously was, in this case and many others, that when they dosed the same formulation made by kinetosol, as by spray drying, we saw a multifold increase in AUC. And once again, and this is in the literature, we published it, it all comes down to congruent drug polymer release and the density of ASD particles promoting simultaneous drug polymer release, whereas with spray drying, the drug, especially with enteric polymers like HPMCAS, the drug is prone to coming out of the particle in the gastric environment or in suspension before the polymer is soluble. So again, we did this with multiple compounds with BI. Here's a couple of studies. That was compound B, this is compound E. This is a primate PK study, once again, showing kinetisol has a significant improvement in exposure. And then here's a more recent case study with Eli Lilly showing the same thing. Spray dry dispersion versus kinetisol, HPMCAS based, we had a 6-fold increase in exposure. And once again, a recent client study where we reduced the drug load and improved the ASD and were, converted from SDD to KSD. So with that, I think since we only have 5 minutes left, I covered the tableting. So just quickly, spray drying, you've got this multi-component process from spray drying to tableting with 2 dilution steps going from roller compaction to final tableting. With kinetisol, there is no densification. We go straight into direct compression of tablets. So there's only 1 dilution step, and typically we dilute with 25 to 20%. So we're getting 75 to 80% ASD load, and that's really critical towards reducing pill burden. So with that, I think I just want to go to my acknowledgement slide. We have a great team here of all these folks on this slide that have helped out in many different ways. I'm just the spokesperson representing a lot of great scientists. So thanks to all of these folks. And then, like I said, most of our case studies are available in the literature. We have numerous publications. So here's the publication history, and reach out to me if you'd like any of these papers. And with that, I want to say thanks, everyone. Here's the contact information. If you're interested in kinetisol development service, leasing opportunities of equipment, or just CDMO support services, please reach out. So with that, I think we have a little bit of time for questions.
Yes. Well, thank you so much for that very insightful presentation. I would like to, at this point, invite the audience to keep sending their questions or comments for this Q&A portion of the webinar. If you're attending via GoToWebinar, you can use the questions window, or for those of you on LinkedIn Live events, you can post your questions in the comments section. Now, I've already received some questions from the audience, so we'll get ourselves started with those. The very first question that I have for you from someone says that earlier you showed a method. They want to know if it's suitable for loading API into mesoporous material, for example, mesoporous silica.
Yeah, so the short answer to that is it requires a little bit of polymer. So we've been able to successfully do it with a ternary system. So drug, mesoporous silica, and a little bit of polymer sort of to bind the composite together. So for mesoporous silica, having to deposit the drug into the small pores of the carrier, solvent processing for those type of excipients is probably preferred, but we've had success using it in ternary systems.
Fantastic, thank you so much. Moving on to another question. Someone wants to know, how does kinetisol fit into a typical GMP facility setup when compared to spray drying systems?
Yeah, that's a great question. I touched on it briefly, but with spray drying, as most of you know, the installation, the capital costs and installation it's very expensive and time consuming. And by contrast, kinetisol is a standalone piece of equipment. It's on casters, as you might have seen in the equipment photos previously. It can roll into any existing GMP facility, plug into the electrical, and supply chill water, and you're up and running at high volume, making ASDs at high volume. So kinetisol equipment can travel. It can go anywhere. It can go in almost any facility. Spray drying equipment is not portable. In fact, as you get up to commercial scale, you've got to construct a building around the dryers. So it's capital intensive. It's time intensive. Bringing up additional spray drying capacity is a monumental task, and it takes years. Bringing up additional ASD manufacturing capacity with kinetisol, it takes a couple of months and a fraction of the cost. So it is much more amenable to incorporation within an existing GMP facility than really any other technology, but especially spray drying.
Perfect, thank you. Next question here is someone wondering why polymer selection is so critical to ASD performance.
Yeah, some folks, the people who are deep in the space understand that drug polymer interactions really depend on the polymer chemistry and the positive interaction between the drug and the polymer, and finding a binding pocket within the polymer that satisfies the drug's need for stability, but also interacts with the aqueous environment in such a way that you're able to stabilize supersaturation. So it's all about the interaction between the drug and the polymer, speciation, the creation of colloidal species that stabilize the drug, but also interact favorably with the surrounding aqueous environment. So, and that varies based on, I mean, the drugs, especially that we're working on now, are very complex. It's not that one polymer fits all. You've got to screen all these chemistries and maybe combinations of chemistries, which we love to do with Kinetisol, combining different polymers to find those favorable interactions, not just drug polymer, but also that complex with the surrounding aqueous environment to maximize the drug available for absorption and to extend the duration of availability so that it can be absorbed throughout the GI tract. So it's polymer chemistry is essential, and that's just from a biopharmaceutical performance perspective. When you talk about physical stability, chemical compatibility, chemical stability, manufacturability, you also have to find chemistries that allow you to satisfy those requirements as well. And that's why I keep stressing the expanded design space that allows one to simultaneously solve these three very challenging problems. Binary ASDs just don't cut it anymore, frankly. They're just suboptimal. So it's polymer, selecting the right polymer, and nowadays it's combinations of polymers. So that's why, that's my perspective.
All right, thank you very much for that answer and for all of the answers, but we have reached the end of our time here today. To our audience, if we couldn't attend to your questions, know that the team at AustinPx will follow up with you, or if you have some further questions, you can direct them to the email address that's up on the screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Now, I've also shared a link to view the recording of today's event in the chat box, which you can also share with your colleagues so they may register for the recording here as well. For those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join me once more in thanking our speaker for his wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks, Ryan.